Hello, everyone, and welcome back to now chapter 8. So, in chapter 7, we spoke a lot about actually cellular restoration. So, mostly these, we talked a lot about how do we actually break down, how do we do catabolism. Specifically, we spoke about how do we break down glucose and specifics more the oxidization, so like glycolysis, and that how we can basically, utilize it to further break it down. Either if we have oxygen as a final electron acceptor, that's then aerobic. It's inorganic, then we use basically anaerobic, or if we have organic molecules that would go into fermentation. So now here we're going to talk about how plants are able to basically create glucose. Because if you guys remember, when it comes to organisms, there are two types of organisms. There are autotrophs and heterotrophs. The autotrophs are organisms that are able to create their own organic molecules. However, heterotrophs are the opposite. They cannot create their own organic molecules. They acquire it by eating other organisms or by other organisms. An example of heterotrophs, as I said before, is humans. We cannot create glucose out of thin air. We need to obtain it by eating. So we eat other organisms, being plants, be it animals or whatever, to obtain our own organic molecules. When the case of autotrophs, these organisms are able to create it, and we're going to be able to talk a lot about this because this is basically photosynthesis. So in the case of photosynthesis, if you want to put this in the scale of evolution of metabolism, it's not super old, but it's not super actually evolved. It's somewhere in the middle because the first things that we spoke about, the first things that we developed actually as primitive organisms to do in metabolism was to break down random bonds to create ATP molecules, and then later on we evolved glycolysis. It was later, only after that, then, when we actually started to see how important glycolysis is for us, we actually developed, you know, mechanisms to create glucose. And that's the first thing that came out were these anoxogenic photosynthesis organisms. So these guys, they create glucose through photosynthesis, but they do not create oxygen as a byproduct. So that's what it calls an anoxogenic. They were actually using H2S because that was what was actually in the atmosphere. So they were using H2S, and they were basically then releasing a lot of sulfur. So that explains a lot. During those times, the atmosphere was heavy in sulfur because all of these organisms that were basically taking in this H2S was releasing sulfur, and that's what made our atmosphere so rich in it. It was later in that organisms started to use actually a different type rather than H2S. We started to use H2O, so water, in a way that we were able to actually break down H2O and release oxygen. And then slowly our atmosphere started to change from heavy sulfur to heavy oxygen because of this change. So now let's talk about these two different types: this anoxogenic photosynthesis and this oxygenic photosynthesis. So the first one, this anoxygenic photosynthesis, is carried out by four different bacterial groups: purple bacteria, green sulfur bacteria, sorry about this, green non-sulfur bacteria, and heliobacteria. So these are the ones that are majorly are doing this anoxygenic photosynthesis. While the oxygenic photosynthesis, they're mostly carried out by cyanobacteria, algae, and all land plants that have chloroplasts. They basically land plants because they're actually living in atmospheres that are rich in oxygen. They need to use oxygenic photosynthesis. Well, these guys here, these actually live in areas that are not rich in oxygen. So that's why they're not, sorry, not rich in water. So they need actually other means to create basically, glucose. Because here they use water to do so, while here they use basically something else. And if you want to have a look at the equation, the simple equation of photosynthesis, specifically oxygenic photosynthesis, it's 6 carbon dioxide plus 12 waters plus a bit of light gives you a glucose molecule, 6 waters, and basically six oxygens. If you think about this, this is literally the reverse respiration reaction. Is literally we're reversing it because in the respiration reaction, we were actually oxidizing, we were basically cutting down um, glucose using oxygen. We were breaking it down to create basically water, carbon dioxide, and basically energy. In this case here, this is light. So now what we're trying to do is basically get the water and carbon dioxide and a bit of energy to create glucose. So it's literally the opposite. Because now this is anabolism. Because catabolism is the breaking, so it's the harvesting of energy by breaking bonds, while in anabolism is the conserving of energies by creating bonds. When you're really trying to basically get like kind of take energy, like get the energy and conserve them in bonds. So that's what oxygenic photosynthesis is. It's basically the opposite, because in the case of respiration, you are doing catabolism. In this case, you're doing anabolism. So let's talk about a bit photosynthesis. Photosynthesis is actually divided into two parts. The first part is light dependent reaction. So where the first part, it requires light. You need light, and what we do with this light is we actually, because we know light can actually be a form of energy. So we use light that's mostly coming from the sun as energy. Because the sun, actually, if you think about it, produces per day more than 1 million Hiroshima bombs in equivalent of energy. So the amount of energy the sun releases per day is equivalent to about 1 million Hiroshimas a day. So it's crazy how much energy is released. And only 1% of that energy is actually taken up by plants because it's quite a lot. So we take that light and the plants take that light and they use that as energy. And they're going to use that energy to actually create ATP and reduce NADP to NADPH. So NADP is literally NAD, but with a phosphate. So it's nicotinamide adenine dinucleotide phosphate. So it's this exact same electron carrier that we spoke in chapter 7, but with a phosphate. So what you do is here is basically we are giving two electrons and a hydrogen proton. So basically, this is the first reaction. It's creating NADPH and ATP. Because then later, this is going to help us to do carbon fixation. So this carbon fixation is where we're using carbon and we're trying to create molecules out of it. So that's what carbon fixation is. And this is light independent, meaning that we do not need light. It doesn't require light. It only requires the ATP and NADPH that we created in the first reaction to actually create synthetic molecules for carbon dioxide. So we create glucose in this manner. So we're gonna talk a lot about this in much more details. But this is just an simplified view of it. So let's talk about where does photosynthesis occur, and specifically where does photosynthesis occur in plants. As we know this, we spoke a lot about this in chapter 4. We know that it happens in chloroplasts when it comes to plants, because the chloroplasts actually contain these very beautiful structures that really do all of these photosynthesis. So so basically, let's do a small recap. We know that the chloroplasts, very similar to the mitochondria, has two membranes. You have the outer membrane, and then you have the inner membrane. Inside the inner membrane, you have something called the stroma. The stroma is the semi-liquid that's surrounding basically everything around here, and it contains all the enzymes necessary to incorporate carbon dioxide into organic compounds using energy for ATP. So you can already say that the carbon fixation already occurs in the stroma, because that's where all the enzymes are. So the stroma is literally the equivalent of the matrix of the mitochondria. So that's how you can actually think about it. Then you have these massive discs, these cycloid discs, which are made of cycloids. So basically, this grana, sorry. So grana is literally it's a bunch of cycloid discs packed on each other. So one of this is called a grana, and each basically has a cycloid structure. And these cycloid is where basically the first part of photosynthesis occurs, the one about the light dependent reactions, because they contain all the necessary proteins and molecules to capture energy and create ATP and NADPH. So the thylakoid, especially this the thylakoid membrane that has this, basically it's a phospholipid bilayer, just similar to a cell membrane. And there you actually contain certain objects that we call pigments, these photosynthetic pigments. And we're going to go into a lot of details about these molecules. These are molecules that are able to capture light and actually utilize or convert that light into energy, and we'll talk a lot about them. So they're actually all found in the cycloid membranes, and they're the ones that are able basically taking up the light as energy, and they're what they're doing is basically creating ATP and NADPH, which then are released into the stroma, and where they do the second part of the reaction, this carbon fixation that's going to use carbon dioxide with the help of energy coming from ATP and NADPH to actually create glucose. So let's go more into some sort of, let's say, recap. This is coming from chapter 35, just about the leaf, the structure of the leaf. So we know that when you look at the leaf, the first ever layer you have is the cuticle. It's this very waxy layer. Remember, this actually that's made of cutin. That this is very a lot of heavy and fatty and these kind of macromolecules there that give this very waxy layer, and it's basically there to protect. It basically helps protect the epidermal cells, and it limits also water loss. So you have first the cuticle, which is shown here. After that, you have the epidermis, which is the first cell layer. It's the first tissue that basically is sticking out in the leaf. So the outermost tissue, which is the epidermis. And then after that, we know you have the stomas, as we spoke about, and the stoma is literally the kind of the mouths of the leaf, which actually happen mostly in the lower part of the leaf or the epidermis, so the lower part of the epidermis. So these are the mouths. Remember? So sorry, no, that's it's this one here. This is the stoma. So these are the mouths, and these are the ones where basically we are getting carbon dioxide to actually help us create photosynthesis. But when we release oxygen, it's released also through the stoma, so through these mouths. And we know these are basically facilitated by these guard cells that we spoke about. Then, in between the two epidermises, you have the mesophyll layers. And in eukaryotes, so basically these are specific types of um, leaves. They basically have two layers of mesophyll. In monocots, no, they have one. But let's just talk about eukaryotes. Eukaryotes, they have two layers, and in one of the layers, basically specifically the upper layer, which is called the palissidae parenchyma cells, is basically it has all of these chloroplasts. That's where all the chloroplasts kind of exists, actually. So it's usually the mesophills that contain so much of the chloroplast, and that's where there actually is the photosynthesis occurring. Actually, it's actually right there. So this is was just a small recap about where is photosynthesis occurring, specifics. So again, let's look into the overview of photosynthesis. So the first thing that happens is you have sunlight. So sunlight is coming out, and basically what we have so this is the cycloid membrane. So these are basically, if you see, these guys over here. So I can go back. These guys. So it's the membrane. In the membrane, you have these very specific structures called photosystems, which we'll speak much more about. And what they do is they actually will collect the sunlight and convert that sunlight into energy. And this energy is really going to, give off the production of ATP and NADPH. And of course, this is a reaction that all happens in the cycloid membranes. And basically you need actually water. You get water and you break water into oxygen. And this is why oxygen is being released. Because, and I'll explain much later why do we break water into oxygen in these reactions. So this is actually almost the opposite as the electron transport system, where we use oxygen and we give it electrons to give actually water. Here, what we're going to do is we can actually break water to release the electrons to help us actually carry out these reactions. So that's why we get oxygen, and this oxygen is released from the stomas of the leaves. So now that we created the ATP and NADPH, now these guys are basically in this layer that we spoke about, the stroma, which has all the enzymes that will basically do this Calvin cycle, which is this whole cycle that basically takes CO2 and is able to actually create organic molecules like carbon dioxide, with the help of ATP and NADPH that you were basically creating here. So all of this, the first part that you're creating ATP and NADPH is light dependent, while this one here, the carbon cycle, the second one that's happening in the stroma, is actually light independent. You just need the energy and so the ATP and NADPH that you get from the first reaction for this to actually occur. So the energy for this reaction is coming from sunlight, while here the energy is coming from ATP and NADPH. So let's do a small little history about photosynthesis and where it started. And I'm going to use the textbook because it's much, much better. Actually, they explain it in a much nicer fashion. So if I go here, yeah. So the first actually clue that came out when it came that the photosynthesis occurs came from this Belgian doctor, Jan Baptiste van Helmont. He was studying. He was very actually curious in finding out like where does the plant get all its nutrients? Does it get it from the soil or does it get it from other basically areas or parts? So what he did is he actually got, if I'm not mistaken, was it the willow tree? Yes, it was a small willow tree, and he put it in, of course, in a soil. So first he actually weighed the tree and the soil, and then he put them together, and then basically he kept providing water. Then after 5 years, he basically weighed the soil and the tree. The tree had grown actually to 74.4 kilograms, so it increased 74.4 kilograms. And then when he looked actually at the soil lost only 57 grams. So then this doesn't make sense. So imagine you're putting a tree and you're putting it in the soil for 5 years. The soil only lost 57 grams throughout that year, but the whole tree gained 74.4 kilos. But is it getting all of that amount actually of food or of that weight if it's not from the soil? So then this was the first clue that like, most probably the willow tree is creating its own food. That was the first thing. However, he had a very bad his reasoning for this was not correct. He thought that, like, okay, it's not coming from the soil, but it's most probably the water he's been feeding past the years. That's what he thought. But then 100 years passed, and then this English scientist, Joseph Priestley, started to see something very interesting. So one thing he used to do is he actually would have this very close container, and he would have a wax candle, and where he would burn it out. So when you burn a candle, you can release a lot of CO2. So that basically in that closed container, you're depleting the oxygen, and there's basically so much carbon dioxide and many other types of gases there that are actually quite deprive oxygen completely. And of course, in that oxygen, you cannot burn because one thing that fire needs is oxygen, actually. One best way to cut out fires is by depriving oxygen. So, here in this small system, when you burned a whole candle, basically you removed all of oxygen. When he tried to actually burn, it never worked to basically burn it again because there was no oxygen. However, interestingly enough, if he put a leaf in that um, container, suddenly the whole atmosphere there changed. There was suddenly now a gas that was actually allowing combustion, and that was oxygen. And he found out that he basically said that vegetation, so basically in this case, the leaves, were adding something back to the air that is vital. And he actually also tested this with mice. Mice couldn't actually live in these containers that you burned these candles because they don't, they, of course, there's no oxygen. So there's no oxygen there, so mice don't live. However, if you put a leaf, now suddenly that mouse can live. So it was actually, adding something back to the air that's vital to animals, which we know to be oxygen. And then later, it was then this physician, sorry, called Jan Egenhaus, that basically he later on, 25 years later, he started to see like, what is this thing that's being released? And he basically, what he started to propose was that these leaves are taking up carbon dioxide and water with a bit of light energy, and it's creating glucose. And it's basically also it's breaking this water, so it's basically using the hydrogens here, and basically it's releasing oxygen. And this oxygen is basically what this something back that to the air, this thing that this Joseph Priestley was talking about, that that vegetation was bringing something back to the air that was vital for animals, which is oxygen. So it was Jan Ingenhouse that actually then came up that actually mixing carbon dioxide and water with a bit of light energy, you started to create glucose molecules and also oxygen gas. This basically that photosynthesis releases oxygen gas. So remember, this is oxygenic photosynthesis. So it was by that time we used to think that photosynthesis was just one reaction. It was only later on, at the beginning of the 20th century, that this black man basically found out that there's two reactions to photosynthesis. One that is light dependent and one that's light independent. So what did he do is he started to measure the rate of photosynthesis. So he started to actually change parameters. So he would increase here, for example, light intensity. So he would increase the amount of light reaching these leaves. He would actually control the carbon dioxide, the temperatures, basically. So that's what he was trying to actually do. And he saw that at low uh, amounts of light, so at low light intensities, if you actually change carbon dioxide and or the temperature, nothing would happen. Actually, as you can see here, so this line over here is when you limit carbon dioxide. This line over here is when you limit temperature, and this one here is the maximum rate, so you don't limit anything. So when here you start limiting the light, so here you limit light here, you start to see basically, sorry, here when the light is limited, that carbon dioxide temperature is not doing anything. It's only actually light is actually being the limit. But then when you have high amounts of light, as you can see here, light doesn't matter anymore. You can keep increasing, but it's nothing's gonna change. It's only then temperature and um, and carbon dioxide that's actually making a difference, a big difference actually. So then he actually found out there's like, okay, there's a reaction where light is super important. Carbon dioxide is not needed, neither temperature. The second one, light is not important because you can keep increasing light and nothing's gonna happen. But temperature and carbon dioxide are actually needed. Then he later found out that carbon dioxide you need it quite a lot, but temperature you can basically, of course, this temperature limitness was mostly because of the enzymes. So if you increase the temperature, the enzymes would work to a certain degree. So he basically saw that the first reaction was very light dependent, while the second one was very carbon dioxide dependent. So this is what actually let us understand that photosynthesis is divided into two reactions: one that is it's called in dependent, and one that's independent. And by that time, we always used to think that photosynthesis only occurs through the oxygenic way, meaning that you have water and that water is being split and oxygen gas is actually released. It was only later on in Stanford, this Hopkins Marine, in the Hopkins Marine Station, there was this CB Van Neal who actually started to see that certain sulfur bacteria do not release oxygen. They started to release actually sulfur in the presence of hydrogen sulfide. So he would add carbon dioxide, this two hydrogen sulfur, and a bit of energy, and you would get a glucose molecule, water coming out, and also you would get this sulfur releasing. And then he proposed this following, uh, let's say, equation for photosynthesis, where you need carbon dioxide, and you just needed a compound, basically an H2A, where A can be basically an electron donor and light energy that gives you the glucose and water and then the 2A. So where this is the electron donor. So this is being broken and the hydrogens are basically donating the electron, basically are donating the electrons. So you're releasing the A. So that's then basically the reaction. So this could be sulfur or this could be oxygen, basically, or something else. As long as basically these H2s are being broken and they're donating the electrons, and whatever attached to it is being released as a gas. So this could be sulfur gas or oxygen gas. So this then really helped us understand that, like, okay, no, there is basically anogenic photosynthesis and oxygenic photosynthesis. So now I'm going to go back here into pigments. So remember, guys, what I was saying in the first reaction here. We need actually, sunlight. We need light. We need basically the energy from sunlight, and we have used these photosystems to trap it and use the energy. So as we said, we need certain objects that are able to have to basically capture this light and use it as energy. And these are called pigments. So pigments are literally molecules that absorb light energy in the visible range. So this visible range I'll be talking about, but pigments are basically are these molecules that are in, of course, the plants that are able to actually get the light and basically absorb it and basically get energy from it. Because again, light is a form of energy, and in specific, it's actually light has photons, these very small particles of light that carry energy. So now let's just talk quickly about um, light. So light is actually, as I said, a photon. So these are small little particles that have energy, and they move in basically kind of these waves. And as you can see, there's something called the wavelength. So the wavelength is the distance from here to here. So basically that it does one turn. So if the wavelength is very big, it means it doesn't have a lot of energy. If these start to basically become very short, so basically the wavelength becomes shorter, they carry a lot of energy. So that's one thing you need to remember. So that's literally light. Light are these photons that are basically traveling in this kind of motion, and they have different energies. So that are very low in energy or very high in energy. So us, when we look at, so in our eyes as humans, we see light as energy. So we are able only to see around this area, which is called visible light. So I have a better, actually, it's this one over here. So that means that the wavelength is basically around 400 nanometers to 740 nanometers. That's the light because we can absorb that light and then basically we reflect it back. So the color is not the absorption, but the reflection back. So this is much more into physics, and you guys will learn much about it in physics. But again, I'm gonna be a bit more here, slower to actually describe this process. So light is basically these photon particles, and they travel at a basically different wavelengths. So if they have a big wavelength, they actually have a very small wavelength, as you can see here. They have a lot of energy. So that's what light is, are literally photons traveling. And then what's how we can basically observe them is if we are able to absorb them and basically reflect it back. Because once we absorb them, we need to basically release it, and release this, of course, as energy. So us humans, we can only actually release, or basically, sorry, or only able to absorb these photons that have a wavelength of 400 nanometers and 740. And then basically we absorb these at these wavelengths, and we basically then release them. So and this release is basically the colors. So this is why we see, for example, blue or red. Depends if we absorb a photon that is basically at 400 nanometers, we will basically release it, and then basically that was blue. This is what we see. So remember, light is not, uh, sorry, color is not light being absorbed, but it's being released. So always remember that. So pigments, so let's go back to pigments. Pigments are literally these molecules that are able to absorb light energy in the visible range, meaning that these are molecules that are able to absorb any photon that has the wavelengths from 400 nanometers to 740 nanometers. That's what literally pigments are. So that's why, for example, when you put actually a plant, in the sun is going to release basically a lot of photons, actually from all of these different types of wavelengths, actually. And that's why plants can carry 1% only is because they will only take this certain basically light. This if basically a wavelength if you get a photon that has 1 nanometer, no way it's gonna get it. Or if it has basically 100 meters, it doesn't have the proper molecules to absorb it. So it's only able to actually absorb here. So as we said, the photons. So what happens is that these pigments, what they do is basically they absorb it. They absorb these photons, and these photons, what they're gonna do is they're gonna crash into the pigment, and when they crash, they could actually release energy as either heat. So this is the transfer of energy. So again, the photons are gonna strike or gonna hit the molecules, and what's gonna happen is that energy is being transferred from the photon to the molecule, which is the pigment. And basically, of course, in this transfer, there's heat always being lost, but of course, energy that's being also transferred. So the molecule is actually going to absorb. So the pigments are going to absorb the energy, and they're going to absorb it by the electrons. So the pigments have electrons, and the electrons by themselves are going to take this energy. So that's what actually happens. So I'm going to show you basically go back to this slide because it's very important for you to understand. So this is basically a atom, as we know, and we know that it has different energy levels. So if we basically give energy to an electron, it's going to go up in energy levels because now it has energy to go up. But once it's up, it has to go back down. And when it goes back down, it releases actually energy. That energy release is actually the color that we see sometimes, by the way. This is the energy that I'm talking about. So what happens here when a photon strikes a molecule, so the pigment, is that the photon is gonna basically hit the electrons. So it's gonna pass it's actually its energy to the electrons. So some of it is gonna be lost as heat because that is the entropy, and some of the energy is gonna be transferred to the electrons. So what's gonna happen is the electrons are gonna get excited and they're gonna start jumping into different energy levels, and then basically later they're gonna go back down and they're gonna release a color, actually a light that's color that we see. So that is basically what happens when it comes to photons and pigments. And these pigments are the ones that are able to actually trap in this light and convert it into energy. But one thing that these pigments actually do is that, as I said before here, they only actually absorb light energy in the visible range. So that means they're picky. They're not gonna get photons that have an energy of this because they cannot absorb it, or the ones that are base are too high. They cannot absorb it. But even in within the actually visible light wavelength, so the 400 and 700, not all pigments will basically be able to absorb all of the energy. So there are mostly two specific types of photosynthetic pigments. You have the chlorophylls, which is divided into chlorophylls A and B, and carotenoids. So chlorophylls A and B and carotenoids, they will actually have a preference to certain photons with certain wavelengths. And I give you an example here with chlorophyll A. So chlorophyll A here, as you can see, so the y-axis is the light absorption, so how much actually light has been absorbed, and this is basically at different wavelengths. So at wavelengths between 400 and 450, it's amazing, this chlorophyll A, but then later, it's just really bad. Around 500, it absorbs it very low. Only later on, around basically 700,